G protein-coupled receptors, or GPCRs, are a receptor class involved in many types of cell signaling. The receptor consists of 7 transmembrane alpha helices bound to a G protein on the interface. The G protein consists of 3 subunits, alpha and a beta-gamma dimer. Alpha is bound to GDP. When a ligand binds, the receptor undergoes a conformational change. The alpha subunit becomes bound to GTP, and the alpha and beta-gamma dimer are freed. They detach and regulate target proteins through second messenger pathways. GPCRs are involved in many pathways, such as the signaling of the taste you sense in your food. 3 of these flavor types, sweet, bitter, and umami, are each detected by specific GPCRs expressed by cells on your tongue. Each cell type is localized to a different area of the tongue into taste buds, or clusters of 100 cells expressing the same receptor. Different subunits are responsible for recognition of different compounds. In general, GPCRs have 3 key regions: an N-terminal venous flytrap domain, a C-terminal 7-transmembrane domain, and a conserved cysteine motif linking the two. The 2 GPCR subunits responsible for sweetness are T1R2 and T1R3. Sweetness comes in various forms: natural sugars, sweet amino acids, artificial sweeteners, and sweet proteins. Sugars like glucose and sweet amino acids bind the N-terminus of both subunits, but artificial sweeteners like aspartame only bind T1R2. The T1R2 and T1R3 proteins are expressed in the taste buds of rodents, but they are indifferent to some sweeteners that humans respond to, like aspartame. Slight differences in the genetic code in T1R2 cause this discrepancy in rodents, but transgenic mice respond to the artificial sweetener just like people do. Sweet signaling is amplified through one of 2 pathways, the first involving CAMP as a second messenger and the second involving IP3. Both of these result in neurotransmitter release and ultimately the sensation of sweetness.
Humans have 25 different bitter taste receptors that are T2R receptors, each with an extracellular N-terminal domain and highly conserved motifs in the cytoplasmic and transmembrane domains. When a bitter ligand, such as caffeine, binds to a T2R receptor, the receptor is activated and activates G-protein gusteucin. The alpha subunit dissociates from the beta and gamma subunits of gusteucin. Alpha-gusteucin then stimulates PDE, which decreases cyclic AMP levels, altering the activity of ion channels and leading to an increase in calcium ions in the cell. Additionally, the beta-gamma subunit of gusteucin activates PLC beta-2 to produce IP3. IP3R3 receptors on the endoplasmic reticulum are activated, causing calcium ions to be released from the ER. The combined effect of these 2 pathways leads to an influx of calcium ions into the cell, which leads to a release of neurotransmitters, causing a response to the bitter taste.
Dr. Kakuna Ikata isolated glutamic acid salts from seaweed and called it umami for its savory flavor. It relies on 3 stimuli: L-glutamate, 5-inosine, 5-guanylate. The taste is strongest when glutamate is combined with a 5' nucleotide. Taste relies on 3 different receptors. The first is a heterodimer of T1R1 and T1R3 GPCRs. The other 2 are variants of mGluR4 and mGluR1. Glutamate receptors are normally found in the brain. These receptor variants have an extracellular N-terminus that is half as large as those found in the brain.
After GPCR secondary messengers cause neurotransmitter release from taste cells, the signal is propagated through normal neural mechanisms. It is thought that the gustatory pathway in the brain is attached to the reward pathway. Whether this reward pathway is activated or not is thought to be the main reason that an organism enjoys or rejects a particular taste. In taste cells, GPCRs help convert molecules that can have complex chemical structure to the signals that are used in the nervous system, so that an animal can develop a preference or aversion to a taste, which performs the important function of determining what an organism will ingest.